No questions. Let me remind you a few things. You can get the YouTube website from this link. There's a link to my syllabus. We're working on chapter 2. I've got the study guide already up. The notes will go up after we're done with the lecture. Remember to do practice quiz. It looks like they're due on the 13th. Why is that one on multiple due dates? I don't remember. And the links for the lab we're doing today are here, and there's the worksheet. You need to click on the worksheet to get the worksheet. Somebody was having trouble with that. And then a link for the infectious disease research project. I've only heard from a few students, meaning a handful of students, who have signed up for our research project. So you need to think about an infectious disease research project that you'd like to work on, and then get it okayed with me. Alright, let's take a look at what we're doing today. We're going to be working on Chapter 2, Chemical Principles. We won't finish it today. We will probably finish it next Tuesday. And then the lab, we'll be working on Lab Module 3 today. Any questions about what we're doing? I think I'll close that down. Alright, so in Chapter 2, we had talked about the carbohydrates. And I believe I talked about this slide here, where I mentioned that carbohydrates are biological molecules with the formula CHO, meaning they have carbon, hydrogen, and oxygen. And they have it in the ratio of 1 carbon, 2 hydrogens, and 1 oxygen. And then you just repeat that if they have more than 1 carbon. Meaning if it's 2 there, you got you put a 2 there, and then you got 2 carbons, etc. Any question about that? So let's talk a little bit more about the carbohydrates. We mentioned that we can break them down into monosaccharides, disaccharides, and polysaccharides. Monosaccharides are called simple sugars. All of them have 3 to 7 carbon atoms, but usually sugars have 5 or 6 carbon atoms, like glucose and fructose. Both of them have 6 carbon atoms. If you talk about ribose, it has 5 carbon atoms. Sugars are sweet molecules. They're molecules that dissolve in water, so we say they're water soluble. The monosaccharides are the simple sugars, 1 unit, and then the disaccharides are 2 of the monosaccharides that have combined together, or 2 sugar units. You can make a disaccharide by dehydration synthesis. Let me blow this up a little bit. Taking 2 monosaccharides and then joining them together in dehydration synthesis, water comes out of that molecule. Let me bring this over here. So water comes out of that reaction, and the water comes from an OH group of one of the monosaccharides and an H group of the other saccharide, forming the water. And then this carbon here, remember wherever there's a point in the ring structure, there's a carbon, binds directly to this oxygen here. So this carbon right there binds to this oxygen, and then we have that carbon bound to this oxygen like that, and that's the disaccharide, which in this case is sucrose, made up of fructose and glucose. Any questions about any of that? Your body needs to do this when it wants to make sucrose, like it's building larger molecules, and then it might take the sucrose and make starch out of it, although I'm not sure how that happens, so we won't talk about that, because starch is only made up of glucose, so I know how you make it just putting the glucose together, but you must remove the fructose, and probably what happens is the fructose goes to the liver and then it's converted into glucose, and we won't get into that. You can reverse this reaction in in hydrolysis, taking the disaccharide, adding water. The water comes in right here. Is that shown down below? No, it's not. The water comes in. Oops, going too far. I'll get this right. I've got it blown up so it's not working well. The water comes in right here, splits this bond, this whatever that the bond is called, covalent bond. And the OH group joins here, and the H group joins here, and splits that, and then we get the disaccharide changing into two monosaccharides by hydrolysis. Any question about any of that? All right, let me shrink that so you can see that. So let's talk a little bit about the polysaccharides. Those are large, complex carbohydrates that are composed of many monosaccharides, and poly means many, so many saccharides. Saccharide just means sugar. So they have many monosaccharides, and they're straight or branched chains, all of the polysaccharides. An example, the easiest example is starch, and it's just a linear chain of glucose, one hooked up to the other, and then they just repeat one after another in a linear chain of molecules. Glycogen is similar. It's a chain of glucose hooked together, but instead of being a linear chain, as most molecules of starch are, glycogen branches. So every once in a while, like on this sugar here, it would branch, and then you get another linking of monosaccharides here. There are some other polysaccharides. We'll only talk a little bit about cellulose. All of these examples are glucose bonded together, but they're bonded together in different ways. And then here we're showing the starchy foods. I'm sure you're all aware: bread, cereals, pasta, rice, and potatoes. They're essentially all they are is starch. I mean, they do have a little bit of other things, but the vast majority of these foods are starch. Now, beans and chestnuts, you might not know this, have a lot of starch in them, but they do also have other food molecules, other biological molecules. Like beans have both lipids in them and protein. I'm sure you all know about proteins. And beans are about 1/3 protein, 1/3 carbohydrates, mostly starch, and then 1/3 fat. And then chestnuts, I don't know the makeup of chestnuts, but they do have a lot of starch as well as a lot of protein. Any questions about any of that? Here's a slide. Let me blow that up so you can see it better. The polysaccharides, it's just for your amusement. Come on, stop that. So you don't need to memorize this slide. You don't need to really know it. I'm just showing you the different polysaccharides. There's starch, linear molecule, glycogen, a branching molecule, but still linear. Cellulose, what it is many linear molecules of glucose hooked together, one after the other. But besides having and this isn't showing well, besides having a linear molecule, it's also, it's branched. It has a side reaction that each, come on mouse, the The dark one is a covalent bond. The side ones, the branched ones, meaning yellow and something, that is a hydrogen bond. So this sugar is hydrogen bonded to that sugar. And then this sugar is covalently bonded to this, come on mouse, this sugar here and this sugar here. So it not only is it a linear chain of covalent bonds, but it's linked to two other chains on the side with hydrogen bonds. And that's cellulose, which is really difficult to digest, and that's why we can't digest it. Any questions about any of that? Is anyone there? I'm not hearing anything, and I don't see anyone.
Yeah, we can hear you.
Okay. Yep, no pictures. All right. Any questions about the carbohydrates? If not, we're moving on to the lipids. Lipids consist of carbon, hydrogen, and oxygen, so the same elements that you can find in carbohydrates, but they differ in that lipids mostly consist of carbon and hydrogen. They do have some oxygen, but oxygen is very rare in the lipid. You got that? So they're mostly carbon and hydrogen, and they have a little bit, just a little bit of oxygen. I'll show you a lipid and then point out the only oxygens in it. Lipids are, most of them, nonpolar and insoluble in water, so we say they are hydrophobic. Literally, that means they fear water. They don't dissolve in water. Now, there is one exception. There is one lipid which is different, and it's actually at least partially polar, and it's hydrophilic, meaning it will dissolve in water. But there's only one exception, and we'll talk about it when we come to it. Most of the lipids, actually all the other lipids, are nonpolar and insoluble in water. They are soluble in other lipids, like in oil, but they're not soluble in water. And you know that if you've ever made vinegar and oil and making up salad dressing. The oil comes to the top and it separates from the water. Lipids are important structural and energy storage molecules in organisms. They play roles in structural components, meaning we have lipids in the structural components of our cells, like the cell membrane is composed of lipids. And then I'm sure you're aware of the fact that we store calories, we store energy as lipids, so that lipids are an important storage molecule. And if you eat too much, what you're going to do is convert whatever you're eating into a lipid and then store it down as a fat. And we'll talk a little bit more about that in a little bit. All right, any question about any of that? Lipids, we can break them into three types. So there are simple lipids, and then there's complex lipids, and then there are steroids. Let's start with simple lipids. Simple lipids are composed of one component called fatty acids, so they are a major component of most, but not all, lipids. And you can build at least some of the lipids, not most, but the majority of lipids, you can build them by using fatty acids, and I'll show you how we do that in the next slide. So fatty acids are a major component of some lipids. They consist of chains of carbons that have mostly hydrogen bound to it. Let me see if I can, didn't mean to move that. Blow this up a little. I'm going to do it this way. So a fairly long chain of carbon bound together, one after the other, with mostly hydrogen bound to the carbon. And you can see on this fatty acid that it does have some oxygen, but only two of the atoms are oxygen, one there and one there. So that lipids do contain oxygen, but it's really rare in the molecule. Any question about that? I'll shrink that down a little bit so you can see that. We can break fatty acids into two types. There are saturated fatty acids, and they have hydrogens, the maximum hydrogens bound to a carbon. Or another way of wording it is that each carbon is bound to another carbon with only one covalent bond. So this carbon here is bound to that carbon here with one covalent bond. There are also unsaturated fatty acids, and they have a double covalent bond between two carbons, and so that they have two less hydrogens bound to these two carbons, meaning this carbon has one less hydrogen, and it's because the bond here with the hydrogen goes to the carbon. And that is a unsaturated fatty acid. You don't need to know the different types of unsaturated fatty acids, but I'll show you what an unsaturated fatty acid looks like. See if I can get that a little better. Wrong one. Here we go. So there's an unsaturated fatty acid. The whole molecule here is a fatty acid, and it's unsaturated because it has a double covalent bond between two carbons. Wherever there's a double covalent bond between two carbons, you'll notice that there's a kink in the molecule, meaning if you were unsaturated, the molecule is straight, and wherever it's unsaturated, where there's a double covalent bond, there's a kink in the molecule. Any questions about that? All right, so that explains fatty acids. Fatty acids can get together with a glycerol molecule. Let me come up here. Here's glycerol right here. And the fatty acid can link together with the glycerol. The fatty acid molecule comes on in dehydration synthesis, and an OH comes off of the fatty acid from the fatty acid part, and an H comes off of the glycerol part, forming the water. And then this carbon here binds directly to that oxygen there, and there you can see is the fatty acid bound to the glycerol right here. Any questions about that? You can get three fatty acids binding to glycerol, one position here, one there, and one there, and this is the most common fat you can find in our diet and in our bodies, and this is called simply a fat when it has three fatty acids bound to a glycerol. And because there's three fatty acids, we call this a triglyceride. Tri meaning three, and then glyceride because it's bound to glycerol. Okay? And like I said, this fat, or triglyceride, is the most common lipid in our diet and the most common lipid in our body, triglycerides.
Quick question.
Uh-huh.
Do we need to learn these formulas for glyceride or triglycerides?
You do not need to remember this formula, okay? You should know that it has three fatty acids and a glycerol, but that's all you need to know. You don't need to know how many carbons there are, etc. Okay? You should know that triglycerides, or fats, are the body's richest energy source. So if you were needing energy, let's say you were stranded someplace near the North Pole, I guess it could be the South Pole too, and you needed to carry food with you to get you from one location to another, and you need a lot of energy to keep yourself warm as well as energy to get from one location to another, you would want to carry with you a lot of fats because they are the body's richest energy source. And it is true that they are the richest energy source in your diet, but that doesn't mean that they're a healthy thing to eat. Okay? Especially if it's unsaturated. It isn't terribly healthy to eat a lot of unsaturated fats. It's much healthier to eat, did I say that right? It's not a good thing to eat a lot of saturated fats. It'd be much easier or healthier to eat unsaturated oils that you can find in vegetable oils, as well as protein and carbohydrates. Any question about that? But if you're ever stranded in some place that's very cold and you need an energy source, you want to take along with you fat, because pound per pound, it's the body's richest energy source. All right, any question about any of that? Let's shrink that down and then move on to the next slide. All right, that might not be showing you everything. Nope. Let's move on to another type of lipid, the phospholipids. And they are the unusual lipid because this lipid can dissolve in water. The reason why it can dissolve in water is it has a head portion, this circular portion, as well as the tail portion. And you'll notice this tail is the fatty acid portion of the molecule, and the head portion has a phosphate group. Well, the fatty acid portion of the phospholipid dissolves in lipids. It is nonpolar, hydrophobic. Okay, any question about that? But the head has a phosphate group on it, which is polar. And not only is it polar, this phosphate group has an electrical charge on it, so it's actually ionic. It's also polar from a little bit from the glycerol molecule right here, and perhaps from the organic group, which is just abbreviated here as an R. Okay, so this polar group can dissolve in water. That makes this molecule very different from all other lipids, because this molecule is a lipid that can dissolve in water. It also makes this entire molecule unusual because one portion of the molecule can dissolve in water, the other portion of the molecule can dissolve in lipids like oil. And there aren't too many molecules that are that way. We call it biphasic because it can dissolve in both water and oil. And you do know one other biphasic molecule. Detergents are biphasic. And that's why when you add detergent to your laundry, when you're washing it in the washing machine, why the detergent helps clean your clothes. The polar part of the molecule can help dissolve the polar molecules in your clothes. Of course, water will dissolve the polar molecules, so you don't need the detergent to dissolve the polar molecules, because water will dissolve it. But the detergent part, which is nonpolar, dissolves the parts of your clothes, the stuff in it, the dirt, whatever, that is nonpolar. So the detergent dissolves that, and that's why it helps clean your clothes. Why adding detergent to the laundry helps clean your clothes. And that's exactly how a phospholipid is. One part can dissolve in lipids, the other part can dissolve in water. Any question about any of that? And here we're looking at the phospholipid in a smaller diagram, the polar head, the nonpolar tail. And when you put this molecule in water, and there's a lot of them in water, they're going to line up like this. We'll talk about these later. They'll line up like this because the tails don't like being around water. So we'll have water up here and water down here, and then the tails will all come together because they like being around other tails because they're nonpolar. Nonpolar molecules like being around nonpolar molecules. And then the heads will face the water. Now, when we talk about the cell membrane, I guess I'll mention it. The cell membranes, the main molecule in the cell membrane is the phospholipid. And you can have membranes looking like this, membranes looking like that, or membranes looking like that. This is a membrane composed of, actually one fatty acid is saturated, one is unsaturated, and that's the way most of our membranes in the people are. If you had an animal that, like a moose, that is in frozen weather, they'll have, at least in the extremities of their, not their feet, what do you call them, their hooves, they will have unsaturated tails in their phospholipids, and that's because these can get cooler and still function, whereas this at cold temperatures it'll become solid, and we won't talk much about that at the temperature a moose is exposed to. And so the moose doesn't want their cell membranes in near their hooves to be like this. And you don't need to know this, I'm just telling you where you can find this. And then in our cell membranes, in most animals, we will have mainly the phospholipid, but we'll have other lipids like cholesterol in our cell membrane. And the cholesterol, when it's in the cell membrane, it gives the cell membrane strength. And so this membrane is stronger than a membrane made up of this. And we have this in our membranes just to give it a little more strength. And we'll talk more about cholesterol later in this lesson. All right, any questions about any of that? Okay, so when you have phospholipids and you put them in water, they'll start forming this, shown here. Let me shrink that a little bit. They'll start forming this, and then this will come and circle around on itself, forming either that or this. We won't talk about this, but we'll talk about this. And let's blow that up a little bit. The fatty acids come together because they don't like to be around water, and then the heads face the water on the outside, and then the heads face the water on the inside. And what does this liposome look like? What does that remind you of? What shape is that?
It looked like an orange.
Well, yes, let's get microscopic.
Looks like a cell.
Yeah, it looks like a cell. And in fact, this is almost what a cell is, because a cell has water on the inside. Cytoplasm is mostly water. Water on the outside. Think of a cell being in the blood. The blood is mostly composed of water. And then a cell membrane that is composed of phospholipids, with the phosphate head being on the outside of the cell, or the cell membrane, and then the head of the other layer of the phospholipids being on the inside facing the water. So this is similar to a cell, because this is what our cell membranes look like. They're mostly composed of phospholipids, and they're a bilayer with the head facing outwards and then the head facing inwards, and then the fatty acid portions being in the inside of the cell membrane. Any question about any of that? Get all the slide there. All right, let's move on to the next lipid. You also need to know the third type of lipids, the steroids and the sterols. Steroids and sterols are very similar. A steroid is a structure with four interconnected rigid carbon rings, meaning this molecule is not very flexible, it's rigid. 1, 2, 3, 4 carbon rings. If there's an OH group attached to the steroid, we call it a sterol. So this molecule, which is a cholesterol, has an OH group on it, and so it is a sterol. And as I mentioned, you can find sterols, specifically cholesterol, in the cell membrane of animals. Whoops, still can't see that. Cholesterol found in the cell membrane of animals, and that cholesterol molecule is in the cell membrane to give the membrane increased strength. Where do you find cholesterol in foods? What foods do you find cholesterol in?
Animal products?
Say again?
Animal products?
Yes. If you eat animals, you'll get cholesterol. And one reason is that cholesterol is found in their cell membranes. You do not find cholesterol in the cell membranes of plants. You can also find cholesterol in the organs because animals make cholesterol, and for some reason a lot of the cholesterol is stored in the organs. And that's one reason why you shouldn't eat a lot of organ meat. What I mean by organ is if you're eating things like heart, stomach, what else? Spleen. Most people don't eat a lot of organ meat, but liver would be an organ meat, and people do eat liver. Some people eat heart, but not many people. I haven't had heart for a long time. Most of us eat steak, and that doesn't have an organ, that's tissue. Why do plants not have cholesterol in their cell membrane? Why wouldn't they have cholesterol strengthen their cell membrane?
They don't produce it
Say again?
because they don't produce it.
That is true, but if they were to produce it, why do they not need cholesterol in their cell membrane? What protects the cell of a plant cell?
Cell wall.
Yeah, the cell wall. So plants don't need cholesterol to strengthen their cell membrane because they have a cell wall, which gives a lot more strength to the cell than cholesterol would. So plants don't have cholesterol in the cell membrane because they don't really even need it. And I did mention that some lipids are composed of monomers, like the carbohydrates. Let's go up here. Some lipids are composed of monomers, like the phospholipids are composed of fatty acid chains and a phosphate group. Oh, come on. And then fats are composed of glycerol and 3 fatty acids. But not all fats are composed of monomers. If you look at the steroids and the sterols, they are not composed of monomers. Okay? Any question about that? So lipids are a little different from carbohydrates. Carbohydrates, if they're not a monomer, they're composed of monomers. And then fats, only some of them are composed of monomers. Some of them, the steroids and the sterols, they are not composed of monomers. Any question about that? All of the other biological molecules are composed of monomers. So carbohydrates, proteins, and nucleic acids are composed of monomers. The lipids are the exception. Only some lipids are composed of monomers. All right, any question about monomers? If not, let's go on to the next of the biological molecule, the proteins. Proteins are essential in the cell structure and the functioning of a cell. So we have proteins that make up the structure of the cell, like you can find proteins in the cell membrane and in other places for structure. And then they perform essential functions in the cell. Like I'm sure you're familiar with enzymes. They have a function in the cell, converting one molecule in a chemical reaction to another molecule. And we'll talk a little bit more about enzymes later. I think it's this lesson. Maybe it's the next lesson. Structural proteins are found in structural components of an organism, like you can find structural proteins in bone, in muscle, and in hair. But you can find them in any structural component, meaning like in the cell membrane, which is a structure of the cell. It'll have proteins in it that make up part of the structure of the cell membrane. Let's see, bones. A lot of the structure of bones is made of the protein, but also it has minerals in it, the calcium, that makes up part of the structure. We have proteins that are important in transporting molecules across the cell membrane, and we call these transport proteins. It'll be a protein in the cell wall. So make this the cell wall. There will be a protein spanning the membrane. It can take one molecule across the cell membrane. There are proteins which are important regulatory proteins, and they regulate some body function, and that could be like a hormone, like a growth hormone, which helps children grow, for example. There are proteins that are responsible for movement. The reason why we can walk is because we have proteins in our legs which are responsible for movement. And if you're talking about a cell that's swimming, it'll be a structural protein which is responsible for movement. So there are proteins that are responsible for movement. Trying to remember, I've got a word there. I guess I've talked about everything. There are proteins that are responsible for some of the bacterial toxins. A toxin is something that is toxic to us, and some bacteria make a molecule which is toxic to us, and it turns out that most of them, not all of them, but most of them are proteins. So some proteins are very, well, you could call them lethal to us, bacterial toxins. But then there are other proteins, we call them antibodies, and they are important to us for defending us against bacterial infections. So some proteins help defend the body, and we call these antibodies. All right, any question about the variety of function, both structural and functional, that different proteins have? As you can tell, proteins are very diverse, and they do many different things for us, more things for us than any of the other biological molecule. The reason why proteins are more diverse than the other biological molecule is because they are composed of monomers, but they're composed of more monomers than the other biological molecule. Meaning carbohydrates are composed of a handful of monomers. Lipids are composed of, maybe a dozen monomers. But most of them are just a handful of the monomers. And then the nucleic acids are composed of 4 or maybe 5 if you're talking about all of the nucleic acids. 5 monomers. Is that right? Well, there are 4 different types in our DNA and 4 in our RNA, and there's 5 total, but I guess there's also 3 other building blocks, and we'll talk about that when we talk about nucleic acids. So let me think about that. Is that going to be 4 or 7? It doesn't matter. The point is proteins are composed of more monomers. In humans, proteins are composed of 20 different amino acids. So the monomers of a protein are amino acids. And you just string the different amino acids together to make the protein. Any question about that? So because there are more building blocks to protein, 20 different amino acids, proteins are more diverse than any of the other biological molecules, I should say. It's also true that proteins tend to be very large molecule. Only the largest carbohydrates compare with the size of a protein, although I guess DNA and RNA are also extremely large molecules, but there's only 2 of them, and there's many different proteins in an organism, and there's only well, there's only 1 DNA molecule. I guess there's 3 main RNA molecules. We won't get into that. The point is there's many different proteins. Proteins are made of amino acids, and amino acids all have this generalized structure. There's a central carbon in the amino acid. A hydrogen is bound to that central carbon, and then on one side of that carbon we have an amino group, that's NH2 right here, and on the other side of that central carbon there's a carboxyl group, a COOH group. And you'll remember from the start of this lesson that the amino group and the carboxyl group are functional groups, and they give this molecule its properties. The amino group and the carboxyl group, and that's why we call this an amino acid, because it has an amino group and a carboxyl group. Also on all amino acids is an R group. That's the side right up here. And the R can stand for anything. That's the rest of the organic molecule. There are 20 different R groups in humans because there's 1 R group for each different amino acid. And humans have 20 different amino acids, so we have 20 different R groups. And here is 1 R group here. You don't need to memorize that tyrosine has this function here, but you should realize that all amino acids have an amino group and a carboxyl group. You should memorize that. When we look at the R groups, there can be different types of R groups. This R group is mostly nonpolar, meaning this CH2 group and this carbon ring here is nonpolar, but this OH group is polar. Let's take a look at the different R groups. I mentioned there are 20 different amino acids. They only differ in the R group attached to the central carbon chain. And these are some examples of the R groups, or the different amino acids. You do not need to memorize these. I'm just showing them to you. You can have a very simple R group, glycine. All it is a hydrogen atom in the R group. Excuse me, the R group's down here. You can have a methyl group in alanine. You can have a branch methyl groups in valine. And these, actually all of these, are nonpolar amino acids. That one too is nonpolar. Let's take a look at the next one. This R group is polar from this NH group and that NH group. This one, only the OH portion is polar. Obviously this portion here of the molecule is nonpolar. Alright, so there's some of the different amino acids. When we're talking about amino acids, you should realize that all amino acids are all amino acids are polar. The amino group and the carboxyl group is polar, and some of the R groups have at least polar regions, like on this amino acid, the OH group of this R group is polar, although that portion of the amino R group is nonpolar. So some of the R groups are polar, some are nonpolar, meaning that proteins are both polar, and at least some of the R groups on some of the amino acids are nonpolar, making a protein both polar and nonpolar. Any question about that?
Yeah, I have a question. Why can't we produce the essential amino acids?
Because humans can't manufacture them from building blocks, and so you have to get them in your diet.
Even if we have all 20 amino acids, we can't make it?
We have all 20 amino acids, but we cannot make some of them. We can't make any of the essential amino acids.
Got it.
So you have to get them in your diet. Okay? Lysine is an amino acid, which is an essential amino acid that you have to get in your diet. I don't have that one shown. All right? So there are some amino acids you have to get in your diet. There are others, I think tyrosine is one, that we can make. I think I'll talk about it later. There's one, I think it's tyrosine that we can make, and I think we make it from tryptophan. But maybe it's the other way around. Anyways, amino acids are linked together by peptide bonds. What you do is you take one amino acid, shown here, and another amino acid, shown here, and you combine them together in dehydration synthesis, meaning an OH group comes off of the carboxyl group of this amino acid, and an H atom comes off of the amino group of this amino acid, and then this carbon is directly linked to that nitrogen. Well, that carbon here is directly linked to that nitrogen. And that happens in dehydration synthesis, making water. And then you can do the reverse in a hydrolysis reaction, add the water and break that bond there. And then the OH group bonds there and the hydrogen bonds there, and then you get the two amino acids. Any question about any of that? Okay, well, there's something you should know, and that is this linkage between the carbon of this amino acid and the nitrogen of this amino acid is rare. It is rare not only in biological molecules, it is rare in the universe. And that's why we call this a peptide bond, that this linkage between carbon and nitrogen doesn't happen that often, even in the universe. And so we call it a peptide bond because we find this bond in peptides. And remember, a peptide is a short chain of amino acids, and you build those amino acids up, you'll eventually get the protein. So a protein is a very long peptide. And so we call that a peptide bond. And when you link all these amino acids together, you get a polypeptide chain that we call a protein. You don't call it a protein until the last amino acid is added. Before that, it's called the polypeptide, composed of peptide bonds linking the amino acids together. Any question about any of that? If not, let's go on to the next slide. When we're making proteins, you link the amino acids together until the last amino acid is added, and then we can say that there are 4 levels of protein structure. There's a primary level, a secondary level, a tertiary level, and a quaternary level of protein structure. Let's watch a video on this. It'll probably take a minute to run. Oh shoot, this uses Adobe Flash. Let's see if I can get it to run.
A protein's primary structure is the sequence of its amino acids. An organism's genes tell the body in which order to assemble the amino acids to make any given protein. Just one switch in this order, and the body ends up with a different protein. Thus, primary structure strongly affects protein function. Watch now as this chain assumes its secondary structure. The chain has bent and snake-like curves as, all along its length, the negatively charged carboxylic acid part of one amino acid is attracted to the positively charged amino part of a nearby amino acid. This helix shape is maintained by these positive-negative attractions. Secondary structure affects protein function by determining which amino acids that are far from one another in the primary structure end up close to one another because of the secondary structure. Now watch this helix acquire its tertiary structure. The amino acid cysteine contains a sulfur atom, and 2 cysteines can form a disulfide bond. In this chain, formation of that bond has the helix folding over on itself. And this fold-over arrangement is the chain's tertiary structure. As just one example of how this level of structure affects function, its presence or absence in a hair protein determines whether a person's hair is straight or curly. To see protein quaternary structure, we need to import additional proteins because this level of structure describes how neighboring proteins align. Watch now as additional proteins form the quaternary structure. The way 2 or more proteins are positioned relative to one another is quaternary structure. This positioning is important for such functions as holding an iron atom in the protein hemoglobin.
All right, any questions about this? Could you hear it okay?
No questions.
I'm sorry?
No questions.
Okay. I think I'll close that. And then what's that? Yeah. Don't know what that is. All right, so there are 4 levels of protein structure. Let's go ahead and just use this. Why am I here? Oh, I hit the wrong button, sorry. The primary structure of the protein is once the protein is made, it's just the sequence, the specific sequence of amino acids, one after the other. And the primary structure doesn't stay primary structure for very long because the nearby amino acids start interacting. They can be attracted to each other. Let's see one that would be attracted like this one can be attracted here. This is can form a hydrogen bond between this oxygen and this hydrogen. And then some will be repulsed from each other. And that we call the secondary structure, the localized interactions between neighboring amino acids that causes localized structure, which there can be, why did I go to that slide? 3 different types. I probably won't ask you anything about the 3 different types. You can have a helical coil, and these amino acids are being attracted to each other. They could be hydrogen bond attractions, but if they're both nonpolar amino acids, nonpolar likes to be with another nonpolar, and so that can be an attraction, and we're just going to call that a nonpolar attraction between amino acids. It's actually called van der Waals forces, and that's why when you put an oil molecule with other oil molecule, they like being together. That's a nonpolar attraction. They can also be a hydrogen bond forming. That's a partial electrical attraction. And they can be an ionic attraction, which would be if this has a positive charge and that a negative charge, that would be an attraction to each other. There can be repulsion, like maybe between this S and that S. There will be repulsion between these amino acids, and that could be they both have a positive charge there, or a positive charge, or this could be, well, they're the same molecule, so let's go with these two. One could be polar, and one could be nonpolar, and then they'll be repulsed from each other. Okay, so those are some of the different attractions and repulsions that can happen in the secondary structure. The important thing to note is secondary structure always is a localized interaction happening between nearby amino acids. So it'll be nearby, neighbors or close to neighbors. Okay? And you can get, like I said, a helical coil. You can get a pleated sheet where, I'm not sure how that goes there. The amino acids going this way, it's hydrogen bonding to the amino acids going this way and that way, and they're hydrogen bonding across each other here, and it just makes a sheet, and then this looks like a protein in a sheet. And there's a third way that it's showing, not shown here, and that is you can have a random coil, and that is where there will be an attraction right here, but then it'll go off that way, and you call it a random coil. And you don't need to know the different secondary structures. But the point you should know, it's a localized attraction between amino acids resulting from localized hydrogen bonds, localized hydrophobic interactions, and ionic bonds. And that could be an attraction or a repulsion. The same with, well, hydrophobic, it'll always be attraction between 2 hydrophobic amino acids. But the repulsion would be if you have a hydrophobic amino acid and a hydrophilic amino acid, they will be repulsed from each other. Okay, any questions about secondary structure? If not, let's go on to tertiary structure. This is the overall 3-dimensional structure of a polypeptide chain. This occurs when amino acids interact with each other, the same as in a secondary structure, but instead of it being nearby amino acids, there are amino acids interacting with each other all over the protein chain. Like in this interaction between here, we have an amino acid right here near the end of the chain, and then one that's fairly near the beginning of the chain interacting with each other. You have the same interactions between amino acids in the secondary structure, meaning you can have hydrogen bonds forming, you can have hydrophobic interactions. Do I have any shown here? I don't have that shown. So we'll just call these hydrophobic interactions. You can have repulsions, like right here, there's maybe a repulsion, a hydrophobic and hydrophilic amino acid. You can have ionic bonds forming. Let me see if I've got one of those there. That might be one here, I can't see it. Where one amino acid has a positive charge, the other has a negative charge. Yeah, that's right here, ionic bond forming between this amino acid and that one. You can have ionic bond repulsion, like if this is a negative charge and this amino acid over here is a negative charge, they'd be repulsed from each other. You have one additional attractive force forming in the tertiary structure, which is not seen in the secondary structure, and that is you can have a disulfide bond forming. Let me blow this up. It happens between two cysteine molecules. So there's a cysteine amino acid over here and a cysteine amino acid over here, and cysteine has sulfhydryl groups attached to it. So it has an SH here, and then an H sulfhydryl group here. Well, the hydrogens come off. So let's take off that hydrogen and take off that hydrogen. And then the sulfurs can bond to each other in a covalent bond. And that is shown here between two cysteine. It's called the disulfide bridge because there's two sulfurs, di-2, sulfide, sulfur, okay? And it only happens between two cysteine amino acids. Any question there? Let me shrink this down. This bond is really strong because it is a covalent bond between this amino acid and that, oh, excuse me, that amino acid. It's much stronger than a hydrogen bond, for example, forming between this amino acid and that amino acid. So this bond can break and reform all the time at room temperature. This bond, once it forms, it's going to be fairly permanent. It can break, but it doesn't break very often. And an example of that is my mother used to get her hair and put it in curls, winding up the curls, the hair in the curl, and then putting the curling hat on, which put steam in the hair and the curls. And then after a half an hour, she'd take out the hair net that put in the steam, and then take out the curls, and then she'd have this nice curl. And then she'd go walking out in the Oregon mist or the Oregon rain, and the water from the rain or the mist would get in the curl, which was formed by hydrogen bonds, and break it. And her curl and her hair would sag and it'd go, ugh. And you'd only need to be in the Oregon mist or the Oregon rain really for only a few seconds, maybe a minute or two, and then the curl sags. So it was an easy bond to break. On the other hand, if she had gotten her hair permed, it forms these disulfide bridges between the cysteine molecules, and this is very long lasting. You go in the Oregon rain and the curl in your perm is fine with the Oregon or Washington rain. In fact, as you know, that'll last about a month from taking showers. So this bond is much stronger than the hydrogen bonds. Any question about that? All right, so can you tell me the difference between a tertiary bond and a secondary bond? If you can't, you should know that, all right? The disulfide bond forming in the tertiary bond is one thing, and the other is that the amino acids can bond to each other all over the protein chain, whereas in the secondary structure, the attraction and repulsions in the amino acids only happen in nearby amino acids. Some proteins only have a tertiary structure, and that would be all enzymes that I know of, like catalase. What's an enzyme you studied in Biology 160? Was it catecholase? I know you did it online, but I'm sure you studied an enzyme in Biology 160. Whatever that enzyme was, it only had a tertiary structure. A tertiary structure happens when there's only one protein subunit to the protein. If the protein has more than one protein subunit to the protein, then we can have a quaternary structure in that protein. And some proteins have a quaternary structure and other proteins do not. They only go up to a tertiary structure. Like I said, all enzymes that I know of only have a tertiary structure. But like antibodies, hemoglobin and insulin, they have a quaternary structure because there's more than one protein subunit that gets together to form the entire protein. Antibodies and hemoglobin are composed of 4 protein subunits that get together. And what the quaternary structure is the attraction and repulsion of the amino acids on one protein subunit being attracted to amino acids on the other protein subunit. The bonds are basically the same that you find in the tertiary structure, including hydrogen bonds, the sulfur di bridges, and then the other weak attractions and repulsions. Okay? Any question about the 4 levels of the protein structure? If not, let's go on to talking a little bit more about proteins. Proteins come in 2 overall shape of the protein. They can be globular proteins, and they're compact, roughly spherical. Hemoglobin's an example of a globular protein. Most enzymes are globular proteins. And then the other shape of a protein is a fibrous protein, and it's thread-like, running like a thread. And collagen would be an example of a fibrous protein. You can have proteins conjugated to other molecules, other biological molecules usually, and we call them conjugated proteins. A combination of amino acids with other organic or inorganic components. They're named by their non-protein component, like glycoproteins, obviously have proteins in it, because they have the name. It's a protein and a carbohydrate. And you can find glycoproteins on your cell membrane. And then there are lipoproteins, which are proteins and lipids. And you can find lipoproteins in the blood. And so remember, proteins are complex and more diverse than all other of the biological molecules. All right, any questions about protein? I'll talk about protein denaturation next if there's no questions. Proteins can denature. That's the loss of the protein's 3-dimensional shape. Usually when the protein denatures, you're breaking the hydrogen bonds, maybe the ionic bonds, and the van der Waals forces holding the protein together. So if the protein takes on a specific shape, let me see if I can see myself here. Sorry. So the protein takes on a three-dimensional shape, and the protein can lose its shape and denature. When the protein is in the incorrect temperature, in the incorrect pH, in the incorrect salt concentration, the protein can denature. All right, any questions about proteins? If not, let's move on to the last of the biological molecules, the nucleic acids. There's two nucleic acids you need to know about, DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. They consist of the monomers called nucleotides. Nucleotides consist of the base unit, which is the nitrogen-containing base, and that's a purine or pyrimidine. We'll talk about that probably tomorrow. And they have a sugar unit, which is deoxyribose for DNA and is ribose for RNA. And the only difference is that deoxyribose has one less oxygen than ribose. And then the third unit of the nucleotide is a phosphate group. So all nucleotides are composed of these three units: a phosphate group, a sugar based on ribose, it might be deoxyribose, and a nitrogen-containing base. Okay? Any questions about that? What am I trying to say here? You can have a different nitrogen-containing base. Thymine is shown here. I know that's not very easy to see. Thymine is shown here, and adenine is shown here. They have a different structure because one is a purine and one is a pyrimidine. This is the pyrimidine, and this is the purine. We'll talk about that more later. Right here. The purines are adenine in DNA and guanine, and the purines have a two-ring structure. The pyrimidines are in DNA cytosine and thymine, and it's a one-ring structure. And in DNA, it's easy to remember the pyrimidines from the purines because pyrimidine has a Y in it, and cytosine and thymine have Y's in their names. So for DNA, just remember that the pyrimidines have a Y in it, and cytosine and thymine are the pyrimidines in DNA. Okay? Any question there? The purines do not have a Y in it. Neither purine or adenine or guanine have a Y in them. Now for RNA, we have adenine, we have guanine, we have cytosine, but we do not have thymine. In RNA, uracil replaces thymine. And uracil is a pyrimidine. The way to remember that is that in RNA, you have to remember that you don't have thymine. It's replaced by uracil, and uracil is replaced by a pyrimidine. And yes, uracil doesn't have a Y in it. That's why I said that rule only works with DNA. But if you remember uracil replaces thymine, you can remember that uracil is a pyrimidine. The other thing I should point out is that A binds with T in DNA, and G binds with C. There will be hydrogen bonding between those links. There will be 2 hydrogen bonds between an AT link and 3 hydrogen bonds. We don't show that. Between a GC link. Here we got it, sort of. So GC has 3 hydrogen bonds between that nucleotide link, and then AT has 2 hydrogen bonds between the nucleotide nitrogen-containing base. So DNA is composed of the sugar, deoxyribose. It is a double-stranded molecule, meaning there's one strand of DNA and a second strand of DNA. And the 2 strands bind together by hydrogen bonds between the nitrogen-containing bases. It's held together by the sugar phosphate backbone, meaning there's a, where do I have that? I don't show it very clearly here. The sugar and the phosphates bind together, connecting the nucleotide bases together. Nucleotide bases are the steps on the double helix of the 2 DNAs. I mentioned there are 4 bases in DNA, and it's a twisted ladder with the bases on the rings. All right, any question about that? If not, I'm going to end here, and I'll see you in the lab. All right? Come on. Come on.